Hi guys, today I am going to videotape a little lab demonstration for you. So in this lab we are looking at the effect of catalase, which is an enzyme in our bodies and all of our cells, on hydrogen peroxide. So hydrogen peroxide, bottle looks like this. You guys are probably familiar with it. It is chemical formula H2O2. This one's easier to see. So H2O2 is hydrogen peroxide. It's actually toxic to our cells, yet our cells produce it. Most things that use oxygen will produce some amounts of hydrogen peroxide, and since it's toxic to our cells, our body needs a way to eliminate it very quickly. So what we have is an enzyme called catalase, and almost every living cell has catalase in it, some a lot more than others. So today we're going to look at what that reaction looks like, and we're going to experiment on changing some different factors and seeing what happens. So I have here some test tubes that I have pre-filled with some hydrogen peroxide. We have, you'll notice, 6 test tubes and only 5 sample materials. And that's because one of our test tubes is going to be a control. So I have them labeled. Hopefully you'll be able to see that when I slide them forward. So the first thing we're going to do is leave this one's empty, so that's our control. So we're going to drop a pineapple in here and see what happens. And I'm going to drop a banana into the next one. Oh, bananas are a little mushy. I'm going to put down in there. All right. We'll put a carrot, a piece of potato, and a piece of liver. Wait a second, drop that down. Okay, so hopefully you can see these. And I am going to push this liver down in here a little bit so it actually hits the hydrogen peroxide. And hopefully you can see that we have a pretty dramatic reaction going on. So let's take a closer look. Our control doesn't really have a whole lot of bubbles. It has a few because hydrogen peroxide reacts to sunlight. So it will start breaking down when it comes in contact with sunlight. So that's why you see a few bubbles in there. So this is our pineapple. And you can see that it's got a few bubbles on the side, just like the control, but it's starting to form this little layer on top. Little layer of bubbles. This is our banana. And you can see it's got quite a big layer of bubbles. Our carrot. Again, it's got some bubbles. It's got some bubble in the glass. And if you look, I don't know if you can see in the camera well, but you can actually see bubbles coming up off of the carrot. I hope that you guys have some hydrogen peroxide around the house. Most of you probably do, and with your first aid stuff or wound care. That you can get some stuff out, pour it in the cup and see what happens, and see yourself up close and personal. This is our raw potato. You can see that it's got quite a little layer of bubbles on it. And this is our liver, which didn't actually get all the way down into the enzyme, or all the way down into the hydrogen peroxide. So you can see as I shake it and mix this up, that it has quite a dramatic reaction, right? The bubbles are all over the place. They won't stay inside the tube, and it actually feels warm to the touch. All right, so again, we're going to compare that to our control, which has very little reaction to it. Okay, so these are all examples of raw materials that are in there. Hopefully you can see the labels on here. You guys can see that the banana reacted a lot, the potato quite a bit, the pineapple, carrot probably the least, and the liver is by far the strongest reaction. Okay, so I'm going to set those aside for a minute. And we'll come back and compare those in a second. And I have another set of test tubes ready to go. So we have a new control. Pineapple, banana, carrot, potato, and liver. And this time the samples that I'm going to use are cooked. So I cooked these by heating them in a cup of water in the microwave for about 2 minutes. So this is the cooked pineapple. Drop that in. I'll make sure they all get down in after we get them all in there. We have the cooked banana, which gets really mushy. We have our carrot. We have our cooked potato. And we have some cooked liver that we're going to drop in there. Looks like this didn't make it all the way in. Stir, get them down in there. Yeah, I didn't get it all. There we go. Okay, so we can see some reactions. This is our control again. Very little bubbles. This is our pineapple. So we have our cooked pineapple and our raw pineapple. Let's see if I can get these to where you can see them on the camera pretty good without a glare. That's a little better. So we have our raw here and our cooked here. And you can see that the raw doesn't have a ton of bubbles in it, but the cooked has even less. All right, so let's compare our banana. So we've got our cooked banana compared to our raw banana. So you can see there's still some reaction, there's still some going on. If I cook them longer, probably wouldn't be anything. All right, so we have our cooked carrot. We have our raw carrot. So you can see that the raw carrot, there's little bubbles. Kind of see the tube looks a little cloudy. That's actually little bubbles all over the sides of the tube. So you can see that there is much more reaction in our raw carrot than our cooked carrot. Still not a ton in either, right? And for our potato, here's our raw potato versus our cooked potato. Again, maybe I should have cooked my samples just slightly longer, but you can see that there's much less. This is a much thicker foam in here. There's much less bubbles, so the reaction has been reduced. And then we have, again, our raw liver, which has pushed the liver back up out of the tube and stopped reacting, compared to our cooked liver. And if I were to take this liver and push it back down into the hydrogen peroxide again, we would see that it's going to continue to make bubbles. And it's filling the test tube back up again. Okay, so again, we have our cooked samples here, our raw samples here, and our cooked samples here. And you can see that in our raw samples, we got a much more dramatic reaction, and some things we had more dramatic reaction than others, right? Our banana was particularly good, our liver is great, and our potato is pretty good too. Carrots and pineapples maybe don't have as much catalase in them. So, question for you, or your inquiry, why is the reaction so much less or even non-existent in our samples that have been cooked versus our raw samples?